Friends and my dear students, myself, Professor Mundhe Sandeep, Assistant Professor and Head Department of Microbiology. Today we are going to see the semester seconds of BSc first year. Already first semester is completed. Due to the COVID-19 disease, we are unable to conduct offline classes. Already you know about the coronavirus. Nowadays it spreaded all over the world, but it is very dangerous problem in India. So please, students, you don't go anywhere outside the home. Stay at home. Wear the mask if you are going outside. Keep the social distance and use the sanitizers, because this is the way, this is the best formula. So you have to avoid the disease from our family. If anybody have any symptoms of COVID-19, just like coughing, sneezing, headache, body pains, fever, so you have to check RT-PCR test, or that is the antigens, and confirm it is positive or negative, because due to which what happens, we can not spread such disease to other family members. This is very important. Okay, so friends, today we are going to see the semester second. In the semester second, there are two theory papers, paper number 3 and paper number 4, and apart from that there is one additional paper, that is the annual practical papers. So one by one we'll see the syllabus of the semester seconds paper number 3rd. The name of the paper is Basic Microbiology and Biomolecules. So the paper, the marks, 50 marks paper. There are 4 units. The first unit, it is a microbial staining techniques. Under that we'll see definition, stain, diacidic stain, basic stain, the exochromes, then chromophores, mordant chromogens, then leuco compound, natural stains, then fluorochromes, decolorizing agent, and counter stains. Apart from that, we'll see the theories of these staining methods. There are different stainings. That's why we'll see the theories of such staining methods. Apart from that, we'll see principles, mechanisms, procedures, and observation of simple staining, monochrome staining, and negative staining. The B differential staining, that we can say also gram staining, and acid fast staining, and C structural staining. Under that, cell wall and PHB staining. PHB means polybeta hydroxy butyrate staining methods. We'll see the details about each and every points in the next lectures. After that, the paper number 2, the viruses, distribution and structure. The first point, we'll see the definition of viruses. Then after that we'll see the bacterial viruses. Bacterial viruses is also called as bacteriophages. Then multiplications, how the viruses they multiply, because in the 2 ways the viruses they multiply. In virulent phage, that is lytic cycle. The development of temperate phage, that is the lysogenic cycles. Because these are the 2 cycle phases. Just like coronavirus, it is multiplying in the human body. How they multiply, how they entered, where they entered, and how they multiply. Multiply means the number of viruses increases inside the body, and once increases the number, what happens? It affects throat, lungs, or any other body parts. Due to which what happens? Due to which the patients are undergoing into the critical conditions. So because coronavirus or COVID-19 disease, it is the part of microbiology, because viruses, whatever the study of microorganisms, because virus, it is the study of microorganisms, that is the main important part of microbiology. After that, we'll see the classification of viruses, LHT systems, because this is the one of the worldwide accepted systems that we'll see. Then distributions and structure of HIV, that is the human immunodeficiency virus. And after that, we'll see the plant, animal and human viral diseases with the causative agent. Just like coronavirus, it is the human viral disease, that also we'll see in the paper. Then in the unit third is the biomolecules. Under that we'll see carbohydrates. We'll see the definition and classification of carbohydrates. Then triose, pentose, hexose, with the example of all these. Apart from that, we'll see the disaccharides. In the disaccharide, we'll see the glycosidic linkage. In the disaccharide, we'll see the example of lactose, maltose and sucrose. Apart from that, we'll see the oligosaccharide. Oligo means trisaccharides. The one example is the raffinose. And after that, the last types of carbohydrates, the polysaccharides. There are two types of polysaccharides, homopolysaccharides and heteropolysaccharides. And last, the biological significance of carbohydrates. All these points are covered in the 12th, but they are in shortly we studied all this part in the 12th class, but in this BSc first year we'll see details about all these things. Apart from that, lipids, we'll see the definition and classification, the types of lipids, that is simple lipids, triglycerides, conjugated lipids, that is the phosphatidic acids and phospholipidic and cholesterol. See biological importance of lipid. And the last, the unit of this paper number third, that is the informational and functional biomolecules. One is the nucleic acids, that is in the under that we will see ribose and deoxyribose sugars, nitrogen bases and nucleosides and nucleotides. After that, we will see the properties, structure and functions of DNA, deoxyribose nucleic acids. We will see the RNA also, ribonucleic acids, properties, structure and functions. And last, what biomolecules of this paper? They are proteins. We will see the definition and classification of proteins. Then peptide bonds. It means the protein, it is made up of amino acids, and each amino acid binds with the peptide bonds, that we'll see. Then enzymes. And last point of this unit 4, that is the biological significance of proteins. This is the syllabus of paper number 3rd. So students, this is the syllabus, and we'll see the details of unit 1st in the next lecture. Till then, bye, take the care, stay at home. Thank you.